Inside your body, right now, a tiny city is alive. Millions of workers are preparing for a critical mission. If they fail, growth stops, wounds never heal, life breaks down. One single cell receives a signal. It's time. Time to grow. Time to copy its secrets. Time to divide into two perfect versions of itself. This isn't science fiction. This is happening inside you every second. This journey is called the cell cycle, and its most dramatic moment is mitosis. But how does a cell split with such precision? That's where the real story begins. To understand mitosis, we must first understand what happens before it. The cell doesn't divide blindly. It follows a strict plan called the cell cycle. The journey begins with G1 phase. Here the cell grows in size, makes proteins, and performs its normal daily functions. It's a checkpoint. If conditions aren't right, the cycle stops here. Then comes the S phase, the most critical step. In this stage, the cell copies its DNA. Every chromosome is duplicated, creating two identical sister chromatids. One mistake here can mean disaster. Next is G2 phase. The cell grows again, checks the copied DNA for errors, and prepares the machinery needed for division. Only when everything is perfect, the cell is allowed to move forward. And that's when the cell enters its most dramatic moment: mitosis. All that preparation in the cell cycle leads to one purpose: mitosis. Mitosis is the process where one parent cell divides into two identical daughter cells. Same DNA, same chromosomes, same genetic information. This is how your body grows, repairs wounds, and replaces damaged cells every day. But mitosis doesn't happen the same way in every organism. There are two main types of mitosis. The first is open mitosis. In this type, the nuclear membrane breaks down completely. Chromosomes are released into the cytoplasm, and spindle fibers attach to them freely. This type of mitosis is commonly seen in animal cells. The second is closed mitosis. Here, the nuclear membrane does not fully disappear. Division happens inside the nucleus itself. This type is common in fungi and some lower organisms. Different methods, but the same goal: accurate division without losing genetic information. Now that you understand what mitosis is, it's time to see how it actually happens step by step. Starting with prophase. Prophase. This is the moment where the cell officially commits to division. Inside the nucleus, something incredible happens. The long, thin strands of DNA begin to condense. They coil tightly and become visible chromosomes. Each chromosome is made of two identical sister chromatids, joined together at a point called the centromere. At the same time, the nuclear membrane starts breaking down. This allows the division machinery to access the chromosomes. Meanwhile, spindle fibers begin to form from opposite sides of the cell, slowly moving toward the center. By the end of prophase, the cell has lost its nucleus. Chromosomes are fully visible, and the stage is set for precise alignment. But chromosomes don't divide yet. First, they must line up perfectly. That critical alignment happens in the next phase, metaphase. After prophase, the cell enters a stage where precision matters most. This stage is called metaphase. The spindle fibers now take full control. They attach to each chromosome at a special point called the centromere. Slowly, carefully, they pull the chromosomes toward the center of the cell. One by one, all chromosomes line up perfectly along an imaginary line known as the metaphase plate. This alignment is not random. It is a critical checkpoint. The cell pauses here. It checks whether every chromosome is attached correctly. Because if even one chromosome is misaligned, the cell risks losing genetic information. Only when everything is perfectly balanced does the cell receive permission to move forward. And when that signal is given, the real separation begins in the next phase, anaphase. From perfect alignment in metaphase, the cell now moves to its most dramatic moment, anaphase. Here, the sister chromatids are finally pulled apart. Each chromatid, now considered an independent chromosome, is dragged toward opposite poles of the cell. The spindle fibers act like powerful ropes, carefully ensuring that each new cell will get an exact copy of every chromosome. It's a race, but one that must be perfectly precise. Even a single mistake could spell disaster. As the chromosomes separate, the cell begins to elongate, preparing its two halves for the final division. And once this separation is complete, the cell is ready for the final phase, telophase, where two new nuclei will form, bringing the division to its climax. After the dramatic separation in Anaphase, the cell enters telophase, the stage of rebuilding and closure. Two sets of chromosomes reach opposite poles of the cell. A new nuclear membrane forms around each set, creating two distinct nuclei. The chromosomes begin to uncoil, returning to their thread-like form. But the cell itself is still one. This is where cytokinesis takes over, the final act of division. The cytoplasm, organelles, and cell membrane split, forming two identical daughter cells, each ready to live, grow, and eventually divide again. And just like that, one cell becomes two. And there you have it, the incredible journey of a single cell. From G1, S, and G2, the cell grows, prepares, and duplicates its DNA. Then comes mitosis, prophase, metaphase, anaphase, telophase, and cytokinesis, where one cell becomes two identical cells, keeping life moving forward. Every second, this microscopic process repeats trillions of times inside your body, making growth, healing, and life itself possible. If this amazing story of cell division fascinated you, don't forget to like this video and subscribe to the channel to uncover more hidden secrets of biology. Stay curious, because science is everywhere.